Hello students, today we are going to discuss central dogma. What is the mean of central dogma? Central dogma basically it is the formation of DNA from DNA and RNA from DNA and protein from RNA, and the whole process is called central dogma. As I have mentioned here, see here. DNA to DNA and DNA to RNA and RNA to protein, right? When the DNA is forming from DNA, this is called replication. RNA is forming from DNA, this is called transcription. While the protein is forming from RNA, this is called translation. And the whole process is called central dogma. The whole process is called central dogma. Let's start the another topic, and this is called DNA replication. What is the mean of the DNA replication? Dear students, this question has been asked so many times in your competitive examination. That's why you have to just remember. What is the mean of the DNA replication? DNA replication means the formation of new strand of DNA from pre-existing strand. This is called DNA replication. It means there is a need of formation of DNA for the formation of the various kinds of the protein. It means the formation of the DNA basically needed for the formation of protein. Let's see how the formation of replication or the process of the replication will take place. Replication. As we know that there is the need of a specific machinery for replication, and this machinery is needed two types of the components. The first component is called enzyme and the second component is called single stranded binding protein. Single stranded binding protein and other energetic material such as GTP, guanosine triphosphate. Okay, now we will discuss about the types of the enzyme, that how many enzymes will be required for the formation of the new strand of the DNA. And then you have to remember the major enzyme is DNA polymerase. Major enzyme is DNA polymerase. But here we have formed a specific trick for memorize the all enzymes which will use in the formation of replication. The first one is called helicase, gyrase, primer, polymerase and ligase. They all belongs to DNA, but this one is RNA, DNA and DNA. DNA ligase is also called the biological scissor. Sorry, not biological scissor. DNA ligase is also called the biological glue because of it will used to join the segment of two DNA or two nucleotides. This one is called the DNA helicase. Second one is DNA gyrase, RNA primer, RNA polymerase. This is the key enzyme for DNA replication, or we can say that this is the major enzyme for DNA replication. By the help of this enzyme, it will recognize the type of the nucleotide. It means we have the pre-existing strand of the DNA like that, and this is the point 5 dash and this is called 3 dash. Okay, the 5 dash end basically where the phosphate group is present and there is a presence of the carbon atom, and 5 dash to 3 dash and 3 dash to 5 dash. Okay, this is the region where the new segment of the DNA will take place from the pre-existing DNA. It will require the DNA polymerase enzyme including the RNA primer. Okay. The DNA polymerase enzyme basically used for the searching of the nucleotides which are present inside the nucleoplasm. Basically, the all kinds of the nucleotide have the different nitrogen bases such as adenine, guanine, thymine, and cytosine. If here is the presence of the adenine, thymine, guanine, or cytosine, then the new strand will be complementary gene for adenine is thymine, thymine complementary is adenine, guanine complementary is cytosine, and the cytosine complementary is guanine. It means we can say that this enzyme plays very important and significant role for the formation of the new strand of DNA from pre-existing DNA. These all enzymes basically responsible for this process and for this purpose, but this enzyme is very most important for the process of continuation of DNA replication.